So we were looking at anticonvulsants, and the classification of anticonvulsants include barbiturates, hydantoins, oxazolidinediones, succinimides, urea and monoacylurias, benzodiazepines, clonazepam, and some miscellaneous agent. The examples for barbiturates, phenobarbital and methobarbital, hydantoins, phenytoin, mefenitoin, ethotoin. Oxazolidinediones, trimethadione, paramethadione. Succinimides, the examples phensaximide, methoxamide, ethosuximide. Urea and monoacylurias, phenazamide, carbamazepine. Benzodiazepines, clonazepam. The miscellaneous agents include primidone, valproic acid, gabapentin, felbamate. So these are the structures of barbiturates, phenobarbital and methabarbital. The difference between these two structures is that phenobarbital is having phenyl group C6H5 at fifth position and ethyl group as the second substituent. In methabarbital, you have two methyl substituents at 5,5 position. Coming to hydantoins, phenytoin, mefenitoin, and ethotoin. So in phenytoin, both the substituents are phenyl groups, and mefenitoin, you have methyl group to the nitrogen and one phenyl group and one ethyl group. In ethotoin, you have ethyl group substituted at nitrogen, one phenyl group at fifth position. So these are the differences between mefenitoin, and ethotoin. Coming to oxazolidinediones, trimethadione and paramethadione. In trimethadione, you have three methyl groups, one at nitrogen, other two at position. In paramethadione, you have one methyl group at nitrogen and one methyl group at fifth position. The second substituent here is ethyl. Coming to succinimides, phensaximide, methoxamide, ethosuximide. You have to observe the difference between rings also. Hydantoins and oxazolidinediones and succinimide heteroatoms are changed. So in phensaximide, you have methyl group to nitrogen and phenyl group here. In methoxamide, you have two methyl groups. In ethosuximide, you have one ethyl group and methyl. There is no substituent at nitrogen. Coming to urea and monoacylurea, phenazamide, carbamazepine. So this is a phenyl ring and this is acetyl group and amide NH C double bond O. If you observe NHCO, NH2 is urea unit. Okay. And observing carbamazepine structure, this is the amide part. Okay. In that, if you observe, this is making it the urea part. Okay. Which is embedded into the ring. This is azapine ring, 7-membered azapine ring. And it is fused to two phenyl groups on either side. And we saw benzodiazepines, that is clonazepam. Okay, we know the general structure of benzodiazepines. We were discussing about benzodiazepines while discussing about sedatives and hypnotics. There we saw the electronegative atom was most commonly chlorine at 7th position. This is 7th position. Okay. Here it is nitro, and you have chlorine to the phenyl substituent. Okay, at the 2' position. So this is the azapine ring. This is a benzodiazepine. This is benzene and this is diazepine fused together, benzodiazepine. Okay, so till here we saw, and we need to see the chemical structures of the remaining candidates in anticonvulsants. Those are miscellaneous agents, right? So what is it? We have trimidone, valproic acid, gabapentin, and pelbon. So let us see trimidone. Is it looking like barbiturate? But here you don't have the third carbonyl. So the two substances present at this place are, one is phenyl group, the other one is ethyl. So remember that primidone resembles barbituric acid but lacks one carbonyl group, whereas barbiturates will have three carbonyl groups. One more here, right? And the substituents include at the position are C6H5 and C2H5, phenyl group and ethyl. If you see the structure of valproic acid, CH3, CH2, CH2, CH, CO, OH, carboxylic acid. Here you have a alkyl side chain, that is propyl side chain. So this is the valproic acid. So how many carbons in the main chain? 1, 2, 3, 4, and 5. So 5-membered carbon chain and carboxylic acid functional group with a side chain that is propyl makes up valproic acid. Fine. Next we'll see the structures of gabapentin and pilbara. So gabapentin contains cyclohexyl ring. Here you have two substituents. One is acetic acid CH2 COOH. Other side you have carbamide CH2 NH2. That is it. Very simple. Gabapentin will have cyclohexyl ring with two substituents attached to it. With the first carbon, one is CH2COH, the other one is CH2NH2. Okay. If you see the structure of albumate, it is also having cyclohexyl ring. Observe the side chain. CH2O CONH. CH2 O CO NH2 carbonyl functional. And to this side the same thing. CH2O CO NH2. So this part and this part are same. Okay. So this is all about the chemistry of anti-convulsants. And let's see the uses. Okay, felbamate is used for the treatment of epilepsy. To treat epilepsy. Gabapentin for partial seizures. Partial seizures. Also treatment of restless leg syndrome. Hot flashes. And neuropathic pain. So these are the uses of gabapentin, and felbamate is used for the treatment of epilepsy. If you see the uses of valproic acid, all types of epilepsy, primidone, which is a potent anticonvulsant, clonazepam, same epilepsy. So you can easily predict the use by seeing the classification, because all these drugs are coming under anticonvulsants. They can be used to treat epilepsies, different types of epilepsies. You have to remember specifically if they have other than epilepsy. Okay. For example, carbamazepine can be used for the treatment of trigeminal neuralgia. Trigeminal neuralgia. And also manic depression. Okay, furosemide, psychomotor epilepsy. Okay, so all fenfexamides used for the treatment of petit mal epilepsy. It is a type of convergence. All the three succinimides used for the treatment of petit mal, epilepsy, and this is the same case with the trimethadione and paramethadione. These two also treatment of petit mal, epilepsy. So coming to phenytoin and other hydantoins, they can be used according to the case in all types of seizures. All the three can be used against all types of seizures. And phenobarbital and methabarbital. Methabarbital can be used for the general seizures, and phenobarbital particular for tonic-clonic. And partial seizures. So these are the uses, particular and general uses of anti-convulsants. Fine. Then we have to see the structure activity relationships of hydantoins. So what is the general structure of hydantoin? So if you number it, first position, second, third, fourth, and fifth position. So this is the general structure of hydantoins and the number. If you observe, it is a imidazole ring. Okay, and it doesn't have double bond, so it will be called as imidazolin. And you have two carbonyl groups at 2,4 positions. So it is a 2,4 imidazolin dione. Okay, this is the general structure. So these are actually close structural relatives of barbiturate acid. Okay, if you observe this unit, they are structural relative to barbiturate acids. But only thing is that these are 5-membered rings, while barbiturate acid is a 6-membered ring. Okay, and in barbiturates we have 3 carbonyl groups. Here you have only 2 carbonyl groups. So this lack of one carbonyl group compared to barbiturate acid will affect in reducing the acidity of hydantoins. Okay. One less carbonyl group than barbituric acid makes hydantoins less acidic than them. Okay, that is the first one. So this is a weak acid. That is one point. So 5-phenyl or other aromatic substance is essential for activity. So fifth position you should have a phenyl or any other aromatic substitute. Okay, for activity, that is compulsory for activity. And alkyl group in the fifth position contributes sedation. Okay, if you have any alkyl, so we have observed CH3, C2H5, most common, right? So what they are responsible for? They are responsible for sedation. Okay. So if you observe the structure of phenytoin, mephenytoin, and ethotoin, you have either CH3, methyl group or ethyl group. So they are causing sedation. Fine. So this is the simple structure activity relationship of Hydantoins. Then if you see the mechanism of action, how they act. Mechanism of action of anti-convulsants. So they are actually sodium channel blockers in nerve cells. Central nervous system, they block sodium channels. So let us assume this is a nerve cell, the membrane, okay, this is cell body. And in order to pass the signal, you have sodium channels. What happens generally, three sodiums comes in or potassium goes out. Okay, sodium comes in, potassium goes out. If potassium comes in, sodium goes out. Likewise, the signal passes from one part to the other. Okay, so during epilepsy convergence, what happens? Rapid firing happens. That means this process is fast forward. So what happens? The rapid and irregular triggering of signal makes people to undergo such conditions. Okay, they will cause rapid movements, rapid body movements. So what we need to do, we have to slow down the signal. In order to slow down the signal, they will block the sodium channels. If sodium channels are blocked, okay, the signal tends to become slow. So that will normalize the situation. Okay. So in the normal situation, generally sodium and chloride ions are high concentration in the outside of the cell. Here sodium are more, sodium and chloride. Inside you have potassium. Fine. So what happens when CUs are there? Sodium ions will accumulate inside the neuron in much concentrations. That will initiate so much synaptic nerve transmissions. Okay, when stimulation is received. The anticonvulsant drugs, what they do, they decrease sodium intracellular ion concentration by activating biochemical process that normally extrudes sodium ions from neurons. So anticonvulsants, what they do, they will extrude. They will activate the mechanism to make sodium ions present inside the nerve cell to go out of the cell. Okay, this is the mechanism of action of anticonvulsants. And if you observe, you have barbiturates also in the category of anticonvulsants. What it is? What barbiturate we have? We saw phenobarbital and methabarbital. Fine. So with regard to anticonvulsant activity, if you observe barbiturates, okay, so the action actually optimum activity is obtained when one substituent at C5 is phenyl. So Optimum activity is good for phenobarbital because one of the substance is phenyl. This is the fifth question, right? And 5,5 diphenyl derivative has less activity than phenobarbital. Okay, so if you substitute both substituents at the fifth position with phenyl groups only, that will have lesser activity than having only one phenyl. And N1 and N3 substitutions here. N1 and N3 substitutions. What they do? They result in increased activity. Okay, so you can increase the activity by substituting nitrogens at 1 and third positions. And 5,5 dibenzyl barbiturate acid causes conversion. So we should not substitute with the benzyl groups. What is benzyl group? Phenyl group linked through CH2 to the ring. If you substitute such a benzyl group, that will cause conversions instead of acting against conversions. Okay, so these are the few points to remember regarding the structure activity relationships of barbiturates with respect to their anti-convulsant activity. Fine. So with this, the unit 4 comes to end.